Dear members of the LOOP Scientific Advisory Board, dear colleagues, it is my particular pleasure to introduce the idea and the researchers behind our initiative to advance precision medicine into the context of metabolism and obesity. The LOOBesity team is comprised of experts in clinical and translational research, imaging acquisition and analysis, epidemiology and study development, as well as molecular science in metabolism and obesity that bridges the main Zurich institutions of ETH, the University Hospital of Zurich, and colleagues from the Technical University Munich. Obesity is the condition that the team has decided to tackle for a number of reasons. With more than 10% of the Swiss population being obese and even much higher proportions in other countries, the clinical evidence is obvious. Of particular importance are obesity-related comorbidities that define the clinical course and patient's outcome. These include metabolic diseases such as type 2 diabetes and cardiovascular disorders as coronary artery disease, stroke, and hypertension. These are highly prevalent in an obese cohort with a much increased relative risk in comparison to non-obese subjects. Nevertheless, obese patients are quite heterogeneous. That includes individuals with a rather low and those with a very high metabolic disease burden. I would like to further introduce hypercortisolism, also referred to as Cushing's syndrome, as a disease state that resembles obesity with particularly unfavorable metabolic consequences, including insulin resistance, dyslipidemia, hypertension, muscle atrophy, and visceral obesity that is also evident by measures of body composition. While overt Cushing's syndrome is a rare disorder, there are prevalent conditions that result in an increase in glucocorticoid burden of affected patients. These include exogenous glucocorticoid exposure, endogenous oversecretion, including mild autonomous cortisol secretion from the adrenal glands, but also glucocorticoid metabolism in the liver and in adipocytes that individual modulate glucocorticoid-dependent pathway activation, finally in target tissues with potential detrimental effects. Glucocorticoid secretion and metabolism are druggable with specific inhibitors of adrenal steroidogenesis, enzymes of glucocorticoid metabolism, and glucocorticoid receptor antagonists. Specifically, inhibitors of 11-beta hydroxysteroid dehydrogenase 1, 11-beta HSD, can counteract peripheral activation of glucocorticoids that has been shown to result in metabolic benefits. Assessment of individual glucocorticoid burden is not trivial and has to rely on patients' history for exogenous glucocorticoid exposure, classic endocrine testing for evaluation of endogenous Cushing syndrome, or urinary analysis as a marker of 11-beta HSD activity. However, quantification of the effects of glucocorticoid on the target tissue itself that integrates all variables of glucocorticoid metabolism has remained to be a challenge.
The main challenge is that you can only assess the ultimate glucocorticoid burden by assessing the receptor activation within the target cells. While this is a huge challenge, this is also a big opportunity because this opens the possibility that a subset of patients suffer from an enhanced glucocorticoid burden, which promotes an unhealthy metabolic phenotype and thus would benefit from treatments targeting this pathway. We did an initial screen in the Leipzig obesity cohort to assess whether such subclusters of patients exist. This we did by measuring the transcriptomics within adipose tissue as a measurement for external factors influencing gene transcription. These data can be used to cluster patients into individual groups, as shown in Figure A. Clustering algorithms defined 5 to 6 independent clusters of patients. If you now look at the transcriptional changes that provide a feedback towards exogenous signal, you can see that especially cluster 6 seems to be dependent on an enhanced glucocorticoid signaling, as basically all of the glucocorticoid target molecules glucocorticoid receptor target molecules were upregulated in these patients. This, of course, is a large opportunity for us to use patients, to stratify patients in the following manner. We have on the one hand, the obesity burden that was mentioned before, ranging from rather healthy to unhealthy people, independent of glucocorticoid burden. And then on the other hand, we have glucocorticoid-dependent pathway activation, which increases the risk of metabolic disorders and thus predisposes patients towards more metabolic disorders. And thus groups B and D might be especially targeted by factors, by interventions that modulate glucocorticoid signaling.
And we are convinced that this now opens opportunity for personalized treatment of obesity as compared to current practice. Current treatment is quite uniform and does not consider the individual profile, and only few receive potent drugs like GLP-1 agonists, likely because there is uncertainty about the precise indication that would make sure that benefits justified side effects and cost. Now, with the characterization of the steroid-dependent pathway activation, we are convinced that we do have an approach that matches well the mechanisms of action of the drugs, as explained previously. And here we see that this approach can be more specific and better identify those who are more likely to benefit from drug treatments, either GLP agonists or HSD1 inhibitors. This scenario leads us now to the overall tasks along the translational pathway and deliverables of the LOOBesity consortium. Firstly, we propose to deeply characterize the obese patient cohorts on clinical grounds by imaging, by molecular phenotyping, with the aim to identify parameters that identify where individuals' patients are on this continuum of the glucocorticoid pathway activation and the metabolic disease burden. Secondly, we will make use of the identified markers to predict the prognosis in terms of metabolic disease burden and the switch between healthy and non-healthy obesity. And thirdly, we will integrate the evidence from our findings and evidence that is available otherwise, like from trials on treatment effects, on benefit and harm outcomes, and we will use established modeling approaches to predict the benefit-harm balance of different treatment options for different individuals. And finally, we will do a randomized trials where we compare this personalized approach to obesity treatment versus the more experience-based way of decision-making. We will make use of existing cohorts and databases. First, by the imaging databases of the University Hospital in Zurich. Second, we can recruit from the Corona Immunitas studies, where more than 10,000 individuals from the general population in Zurich participated. We can recruit participants for the cross-sectional studies, and we may also recruit there for then the longitudinal study for the Zurich obesity cohort that is already running, and that will provide the data for the longitudinal analysis.
The characterization of enrolled obese individuals is done by clinical phenotyping, imaging, and molecular phenotyping.
Especially the molecular phenotyping uses completely novel approaches, integrating data on cellular as well as individual cell changes by studying promoter occupancy, in this case focused on glucocorticoid receptor dependent pathways. For this reason, we have also included Henriette Uhlenhaut in our consortium, who's one of the world-leading experts on glucocorticoid signaling in target tissues.
Why imaging and why MRI? MRI allows a non-invasive and radiation-free quantification of different fat depot, like the subcutaneous and visceral fat. Different studies already showed the image-derived body composition is capable to define certain phenotypes. But the drawback of all studies is the long-lasting examination time. Our goal is to provide a fast and robust MR scan, which is independent from scanner type or field strength. Just by omitting body calls, we can save more than 10 minutes in patient positioning. Initial results show that the image quality remains almost equal with and without calls, enabling further post-processing and patient characterization. Imaging must also be simple and reproducible, like standing on a scale. scale. But instead of number, you will get detailed information about your body fat composition and therefore your health.
The characterization imaging will use a robust and fast acquisition and aim to integrate molecular information. We will get our ground truth about glucocorticoid dependent pathway activation from the molecular signature and aim to build imaging models that takes into account not just images or individual pixels, but also the entire image information, including the amount of fat, but also the distribution of fat depots. Use it in a deep neural network and predict the pathway activation. Our goal is to avoid acquiring the molecular signature for every patient and predict the pathway activation directly from the images.
We will then proceed based on all the insights from task 1 to develop prediction models for outcomes in obese patients, as well as prediction of treatment response. We will do so in 3 steps. The first steps will be to identify the best candidate predictors, and we will go to the imaging database and the UK Biobank to do so in order to save our LOOBesity cohorts for then step 2 and 3, which is the development of prediction models and the validation of the prediction models that will include advanced internal validation techniques as well as external validation. Task 2 is important also to bring into task 3, where we will use mathematical modeling to estimate the benefit-harm balance of specific individuals under different treatment scenarios. So with multimodal treatment versus adding a GLP agonists or an HSD1 inhibitor. So we will use established models to estimate the probability that one or the other treatment is better for particular individuals. To bring it to the bedside, we will develop personalized patient decision aids. These are tools to inform patients and the healthcare providers about conditions, the treatment options, based on the best available evidence, clarify preferences of patients, and then provide, based again on the mathematical modeling, an estimate of the personalized benefit-harm balance estimate. Of course, the decision will then still be up to the patient and healthcare providers. We will then expose this new personalized treatment approach to randomized controlled trials. This is not commonly done in the personalized medicine field. So we will randomize participants to the usual treatment decision-making and treatment, which again is quite uniform, versus the personalized treatment decision-making, where we hope to make more precise indications, in particular for those who benefit from drug treatments. Based on the best available evidence and knowledge we have up to this point, we expect to increase the conversion from non-healthy obesity to back to healthy obesity after 12 months, from about 25% with conventional treatment up to 40% with the personalized approach.
How will we handle the data? We will have data from the obesity cohorts, the clinical data, the imaging data, as well as the molecular data, which will be pseudonymized in the LOOBesity database as a part of the larger LOOP database. This will help us to run the AI algorithms that have been mentioned before in a secure environment to conform with any regulations regarding human research. Ultimately, our data will be part of the LOOP clinical data store, where LOOP-related studies and institutions feed in data to allow other projects in the area of metabolism and obesity, but also any study related to this field, to utilize the data and thereby generate new knowledge on patient benefit-harm risks and possible new treatment modalities. With this, I'd like to thank you very much for your attention, and we are looking forward to seeing you in a few weeks.